Welcome to Silent Vibes. In this video tutorial, I am going to discuss about the topic vectors. That what is it? What are the types of vectors? What are the characteristics of cloning vector? And what is carrying capacity? And what are the properties of the idle vectors? So vectors can be of three types. It can be cloning vector, shuttle vector or expression vector. In the cloning vector, it has the small size. It has the origin of replication, that is the ORI region, and it has the high copy number. Selectable and screenable marker are also used in the cloning vector, and regulatory elements are also used, and multiple cloning site and unique restriction sites are also be used in the cloning vector. So what is unique restriction site? It means the location site of each restriction enzyme. Unique means mostly one, but may be greater than one. And what about multiple cloning site? It means the site of multiple restriction enzymes. Means any enzymes can be used, such as HIND3, BAM-H1, ECO-R1, etc. Then in shuttle vector, it has more than one host. And in expression vector, protein synthesis can be done. And ribosome binding site are also be used in the expression vector, and start and stop site, and it has the low molecular weight. Then what are the characteristics of cloning vectors? Here all the cloning vectors are carrier DNA molecules, and these carrier molecules should have few common features in general, such as it must be self-replicating inside host cell, it must possess a unique restriction site for restriction enzyme, and they should easily isolated from host cell, and it must possess some marker gene such that it can be used for later identification of recombinant DNA, and usually an antibiotic resistance gene that is absent in the host cell. And introduction of donor DNA fragment must not interfere with the replication property of the vector. Then the vectors and carrying capacity. So what is carrying capacity? It means the highest limit of the gene of interest which can be carried by vector. And if the vector M3, it has the E. coli host and its insert size is 1 to 4 kb, and if it has the plasmid vector, then its host is E. coli and its insert is approximately 8 kilobase pair, and if it has the lambda phase, its host is also E. coli and its insert size is 5 to 25 kilobase pair. Then these are the vectors, and mostly the host is E. coli and its insert size is varies from 35 kilobase pair to 45 kilobase pair, or it can varies up to 2000 kilobase pairs. And host can be yeast, E. coli or mammals. It varies according to its vector. Then what are the properties of host? A good host should have the following properties. It is easy to grow and transform. It do not have restriction and methylase activity. It also do not hinder replication and recombinant vector. It is easily retrievable. So what are the properties of host? A good host should have the following properties. It is easy to grow and transform and easily retrieval from the transformed host, and it has deficient in recombinant function so that the introduced recombinant vector is not altered, and do not hinder replication and recombinant vector, and it do not have restriction and methylase activity. So these are the properties of a good host, and various hosts are used in the recombinant DNA technology depending on the goal. For example, bacteria, yeast, plant cells, animal cells, and the whole plants and animals. Then the prokaryotic systems such as Escherichia coli are commonly used due to various advantages, like they have the compact genome and easy to transform, and they can be well studied expression system, and they are versatile also, and they are widely available, and the rapid growth of the recombinant organisms with minimal equipment. So these are the advantages of the prokaryotic system. Then we should also know about the properties of vector. So a vector is an autonomously replicating DNA molecule designed from a plasmid or phage DNA to carry a foreign DNA inside the host cell. The transformation vectors are also of two types. That is, the cloning vector is used increasing the number of copies of a cloned DNA fragment, and the expression vector is used for expression of foreign gene into a protein. And if a vector is designed to perform equally in two different hosts, then it is called as the subtle vector. Then we should also know about the properties of the ideal vector. So a good vector should have the following characteristics. It is autonomously replicating, and it may contain a scorable marker, and it contain at least one selectable marker, and it has the presence of unique restriction sites, and it have multiple cloning sites, preferably small in size and easy to handle, and it has the high copy number, and they are autonomously replicating, that is, they have ORI region, that is the origin of replication, and they contain at least one selectable marker, example the gene for antibiotic resistance, and the relaxed control of replication to obtain multiple copies, and the presence of approximately regulatory elements for expression of foreign gene. And the selection of a suitable vector system depends mainly on the size, limit of insert DNA, and the type of host intended for cloning or expression of foreign DNA. Then we should also know some of the basic terms such as what are plasmids, what are the phage vectors, and the term cosmid, phagemid, phagemids, and the artificial chromosome. One should also know about this. So the term plasmids are the circular molecules that exist independently of chromosomal DNA and can replicate autonomously. And the plasmids carry one or more genes which mostly code for useful characteristics of host, and all the plasmids have sequence that can act as the origin of replication, and the plasmids of different sizes and possessing different copy numbers are also present. Then the term phage vectors, as it consists of mainly DNA molecules, it carries large number of genes and are surrounded by a protein coat which is known as the capsid, and they can be used as vehicles for carrying DNA insert after modification to remove the pathogenic genes and minimizing the size. And the term cosmids, what is it? It is the hybrid between a phage DNA and the bacterial plasmid, and they have the cos sites which are essentially required for packaging the lambda into the phage protein coat. And what is phosmids? As phosmids are cosmid like plasmid, but they are based on F plasmid, and the phagemids are what? Phagemids are plasmids having a part of M13 genome. Then the artificial chromosome, what is it? As the artificial chromosomes are artificially constructed DNA, construct used for transferring DNA. Then the vectors such as plasmid, phage, cosmid, BAC, that is the bacterial artificial chromosome, and YAC, that is the yeast artificial chromosome, and MAC, mammalian artificial chromosome. It is derived from following sources, and it has following applications also. So let's discuss what is it. As the plasmid vector, its insert size is less than 15 kilobase pair, and its source is bacteria, and it is used for subcloning and downstream manipulation, the complementary DNA cloning and expression analysis. And the phage vector, it is 5 to 10 kilobase pair. Its insert size is 5 to 10 kilobase pair, and it is derived from the bacteriophage lambda, and its use as its genomic DNA cloning and complementary cloning and expression library. Then the cosmid vector, its insert size is 35 to 45 kilobase pair. It is derived from the phage containing bacteriophage lambda cos sites. As the genome library construction, it is used here. And BAC, that is the bacterial artificial chromosome, its insert size is 75 to 300 kilobase pair, and it is derived from plasmid containing ORI from Escherichia coli F plasmid, and it is used in the analysis of large genomes. And what is YAC? It is the yeast artificial chromosome. Its insert size is 100 to 1000 kilobase pair, that is 1 megabase pair, and it is derived from the Saccharomyces cerevisiae, centromere, telomere, and autonomously replicating sequence, and it is used in the analysis of large genome. YAC transgenic mice also. And what is MAC? It is the mammalian artificial chromosome, and its insert size is 100 to more than 1 megabase pair. And mammalian centromere, telomere and origin of replication are its source. It is derived from the mammalian centromere or telomere, and it's used under the development for the use in mammalian biotech or human gene therapy. Then the PBR322 vector. It is widely used in Escherichia coli cloning vector, and it is created in 1977. And here in PBR322, here the P stands for plasmid and BR stands for Bolivar and Rodriguez. And PBR322 is 400-361 base pairs in length. PBR322 plasmid has the following elements such as REP replicon from the plasmid PMB1, which is responsible for replication of the plasmid, and the next ROP gene encoding ROP protein. And the ROP protein are associated with stability of RNA1 RNA2 complex and also decreased copy number, and the source of ROP gene is PMB1 plasmid. Then the next is TET gene encoding tetracycline resistance. It is derived from PSC101 plasmid. And the last is the BLA gene. It encodes beta-lactamase, which provide ampicillin resistance. Its source is transposon TN3. So here in this video we concluded all the vectors and all its properties also. So thank you for watching this video. If you like the video, please do like, share and subscribe our channel. And further vital topics as highlighting the CSIR syllabus will be coming. So stay tuned for the upcoming videos.